better an echo and so on. Okay, so anyway, I started, if you didn't hear me, by thanking people for inviting me. And this is an important point. The representation of the world is a tough problem. We're, in neuroscience, working hard on that. A lot of progress has been made. However, much less progress has been made in understanding how we represent actions, whole sets of actions. How is it, for example, that we can intend to do something and then do it? This is a very big problem, and I think one of the biggest surprises that's come in the last while is the realization that the deep brain has a lot to do with this function. You're looking at the brain of a normal person, but this, man, this is technically challenging. There's a tiny little red spot. You see it? I've got it here right above action. So if you look over here, this is the deep part of the brain I'm talking about, just the tip of the iceberg, really, of the striatum. The pink is the dopamine. You saw slides like this yesterday showing this striatum of the basal ganglia. And as Parkinson himself pointed out a long time ago, the main problem, or core problem in this disorder, is translating a will and intent into an action. So this is a very deep issue in neuroscience. And this slide makes the point that it's not just Parkinson's that this deep brain is related to. I'm having a lot, this is not a very bright pointer, but there is another one?
Yeah, well, it's like a path to the right.
To the right.
To the right, yeah.
Ah, let's see. Oh, that's much better. Okay, so it's not just Parkinson's. A number of the so-called motor plus disorders have at their root dysfunction in this system of the basal ganglia. The little cartoon shows you the neocortex projecting down, in this case to the striatum. And there are classic outflow pathways long thought to release or inhibit movements. And we now know that actually this system has been implicated in a number of neuropsychiatric disorders quite clearly and surprisingly. It seems like every few months we're learning more about the involvement of the striatum and the basal ganglia even in mania, depression, schizophrenia, let alone ADHD, OCD, Tourette. And it turns out that this system in normal people has a lot to do with how we make habits and whether we get repetitive movements going in our behavior to the point that they might be disruptive, and then addiction, which you heard about a little bit from Linda. So the question is, how on earth can the basal ganglia do all this? And one part of an answer is that in a way the brain has played a trick on us. The basal ganglia, which are well known to project downstream to the so-called CPG, central pattern generators of our hindbrain and spinal cord, they also project up, and interestingly, they project up to the frontal part of the cortex, including not only the motor and premotor areas of the cortex, but this huge prefrontal cortex that we have, and I'll come back to that part of the cortex in a minute. Now, many of the people in my field and allied fields kind of agree that the basal ganglia and the neocortex are partners. We're not sure who's the boss, but there are great looping connections that link them via a bunch of outflow pathways. Interestingly, the dopamine that Linda Buck mentioned is coming in to modulate these loops, and they're thought to help us select what to do, select which action to emit. And in fact, that dopamine itself and its cells, the neurons that house the dopamine, turn out to carry information that's predictive of the cost or benefit of that action. So this is an amazing system, a selection system that's modulated, of course, not only by dopamine, but by many other regions, modulators that can set the decision-making that winds up as selecting what to do. Now you can imagine that if you had such a system and you played those loops over and over, round and round, then what might happen is that you begin to do the same thing over and over again. And this is really the core of what I want to talk with you about. That through these very diverse, very multiplexed loop systems, somehow value, positive or negative, gets attached to sequences of behavior. So we decided we would go after this, and our hypothesis, as I have shown you, is that this really is a learning system. That's what we think at their core the basal ganglia are. So we decided we'd have to put a lot of electrodes in here, and we do have a fair number of electrodes when we record in the striatum. We have to record when animals are performing complex sequences as they form habits. We need to manipulate value. We have to perturb the loops. I don't have time to talk about, we're doing a lot of work now perturbing genes or engineering mice. But we have to do these things in order to try to figure out what's going on. And so we picked the simplest, we thought, naturalistic behavior to have little mice and rats do a T-maze task. And in the T-maze task, they get a little signal that tells them a door will open, so we can note the initiation of their movement. Then they get a cue which tells them to go right or left. They do it correctly, they get chocolate or some other reward. And we can record day by day, and the trick of this is we start out when the animals don't know anything. They're familiar, so they aren't too frightened of the maze, but they haven't learned the task. So every single day we bring them to the lab, we record from the neurons, and we listen to how they respond. And when we started this some years ago, we were really quite surprised by what we found. Now what I'm going to show you is a kind of heat map type plot of neural activity as the animals run down the T-maze. So here's the warning cue, here are the cue, the turn, goal. And red means a lot of activity. I'm going to show you the training days going down here. And you can see that during the run, here's the start all the way to the goal, the neurons in the striatum, this is the sensory motor striatum, it's an important point, they're very active. They really, they turn on. And here are the first 5 stages of training. So by this stage, animals have reached 72.5% correct, a typical chi-square criterion, that they had finally gotten the task. And you may notice a little something funny going on here, but what surprised us was this. So if we kept training, and now I split the plot to show you what in the jargon is called overtraining, really grinding in this training and learning through all these successive stages, you can see that the activity during the runs gradually faded away, not entirely, there are little what we call expert neurons still firing away, but most of the activity wound up at the beginning and the end of the maze runs. So this began to suggest to us that maybe as we over and over do a task that's rewarded and we get better at it, we finally come to the point of packaging that behavior. We in effect, we or the basal ganglia or the striatum, the loops, they decide this is good enough to keep, I'm going to put this behavior in a package so I can do it again the minute I have a cue. And anybody who has ever done something like, any kind of little habits you have, you know that a cue can instantly bring out the whole behavior. A typical example is the person who's tried to stop smoking, used to keep the cigarettes over here, looks over here, and you know the game is up because the cue's so powerful. So it turns out this start and end, or beginning and end bracketing pattern is true in non-human primates. This was a study I did some years ago with now Takafuji when he was in the lab. And you can see in this population plot, big beginning and end responses. These actually are responses to the saccades that the monkey was making looking around the screen. And these have now been found, and we do it in the lab now with lever press tests. So this is a very general phenomenon. When we do a behavior that we wind up finding valuable. So the idea we're working on now is that in the whole stream of behavior that we go through every day, we have episodes of behavior. The episodes are eventually bracketed, as I said, by this amazing basal ganglia system. And the interior of this whole package is less clearly represented, at least in the way we have so far recorded, although I might add there's lots of oscillatory activity inside. And so we wanted to know, gee, you know, how could this work? What do you do with a bracket when you don't know what's going on in the middle? So Kyle Smith and I have done a series of experiments, and I'm going to try to keep my talk pretty much restricted to these experiments. We're recording in that prefrontal cortex I pointed out in the medial part and the sensory-motor striatum at the same time in similar tasks. And let me show you what we found. So we're recording in two loops. One of them is limbic, that's here. We're recording infralimbic cortex. And the other loop is a sensory-motor loop. This is, of course, a new set of animals. And here's what we're finding. In this new set of animals, you can see that bracketing pattern form again. Here's the overtraining period. And we were really stunned because in the medial prefrontal cortex that we thought would be alive with excitement, what happened was it was sort of a little bit alive, but it didn't form the bracket during the training period. And then about halfway through training, and you can maybe see it more clearly when I put these plots side by side, as the habit really crystallized, all of a sudden, we found the bracketing pattern in the limbic cortex. We were very, very surprised by this. So the idea this suggested was that, gosh, maybe it's this part of the limbic cortex that actually is keeping track of the habit as it's developing. And there's a way to test for that. But first of all, let me show you, we needed a way to find out whether the animals were really doing things kind of semi-automatically, or whether they were running these mazes deliberatively. And it turns out when an animal, I'll show you later in a video, when the animal goes down the maze and he doesn't really know it too well, he does the following. Here's just one trial of tracker data. So the camera's up on top looking at so you can see him look one way and then decide to go the other way. And then here is it a little later, the animal knows the task very well, and there aren't any of these so-called deliberative head movements. And in fact, we use those as a kind of surrogate for how much decision-making the little fellow was doing on the fly. And this y-axis here in this plot shows the amount of deliberative head movements, or their number. And here is training going on. And you can see they fell very sharply from early acquisition here, then the middle of the training, and then late training. And these are line plots of the neural activity. Now neural activity is in the y-axis. And you can see early training, middle training, and then finally in the overtraining, this beginning and end activity came. So we had a way to begin to say that, gosh, as that pattern forms, that's just when the animals appeared less deliberative. So could it be that we're catching part of this shift, which is so important in our just human everyday behavior? If we had to do everything deliberatively, we would be in trouble. So there's a way to test whether some of these ideas could be true or could have some merit, and in particular, could we finally get at this question of value? And the way that people do this in psychology, and we simply copied, was they have the animals, for example, in our case, we had 2 rewards. The little rats liked them equally. And so we'd run them in the maze test, then take them back to the home cage at night or the afternoon, and then let them have as much, for example, chocolate as they wanted. Take the chocolate, take the chocolate. And at the end of about an hour, we unfortunately would give them a little shot of lithium chloride in their tummy, and what this does is make them feel sick to their stomachs. So we call this the so-called bad restaurant experiment. I felt so guilty that we did it. But the issue is, if you got a favorite restaurant, you go there every Thursday night, but one time you get sick, will you go back next Thursday? And the probability is, if you've gone there for years and years and you always see your friends, you'll go back. But if you've never been there before, and you have one episode, you may not. So this is what psychologists use to test whether animals have habits. And Kyle Smith and I did this. And it's very important that I emphasize that. And so here's what happens. If animals have only been trained to criterion, and that was the top part of those plots, so they just barely know the task, then if you devalue the reward, as I told you, and you stick them back in the maze, but it's a so-called probe day, so there aren't really any rewards there at all that day. You just check whether they'll run one way or the other. What you find is they will not go back to the bad restaurant. But the animals that have been heavily overtrained in blue, they are highly habitual. So the buzzer comes on and says, go to the chocolate that you know good and well makes you sick, and they keep going to the chocolate. So this gave us a chance to look in the brain and find out whether either of these regions would have different activity when we manipulated the value. And the answer was we thought, quite interesting. Look at the dorsolateral striatum, the sensory-motor striatum, solid as a rock after the devaluation protocol. We can't tell the difference between these. But in that limbic medial prefrontal cortex, that beautiful split pattern, beginning and end pattern, was almost fully hidden when we devalued, and it happened fast. So what we're thinking is that if we do develop a habit, and these systems do operate, then if that habit is strong, it's stuck in our basal ganglia. It's just stuck in the striatum. But in the neocortex, it can let it go. It'll pick up some new kind of activity. So the neocortex, this medial prefrontal cortex, we think is very flexible. This is good news for us. I don't know if the striatal solid rock type activity is good news, however. So we decided the best thing to do would be to go to that flexible cortex and perturb it. Namely, try to shut it down. Of course, now with optogenetics, you can do that. And so with halorhodopsin injected virally in here, you can see the details here, we perturb. And how do we do that? And this is critical. Here's a little cartoon to show you. Here's the maze. We only turned on the perturbation as the animals ran down the maze. Not ahead of that during the planning period, not after that when they got their reward, not even after that when they were kind of rehearsing what they'd done or thinking about it, just as they ran. And so here are the normal animal data I just showed you a second ago. And here's what happens if you shine that light just during the runs. What happens is even overtrained animals look as though they never were overtrained. The habit has been blocked. And it's amazing how rapidly this can... I just wish you could... Can you see this? It's really fun. I hope the light's low enough. Can you see it? Oh, good. Okay. To me, it's just a glare. So this is... you can see we have a very sophisticated lab. But it's a fun lab. And then here is... now we're going to shine the light. So... just as we open the gate. Oh. Maybe I'll have to do it this way. Oh, that's too hard. Here we go. Here he's going to run. See what happens. So it's just amazing to sit with these animals because it's as though they just don't know what to do until they finally do something. So I want to make a couple of points about this. We can, in real time, that's the interesting thing, in a matter of a couple of seconds, prevent the expression of a habit that is very, very well ingrained. We could do this by perturbing originally, actively, a very tiny piece of the cortex, medial prefrontal cortex. The blockade is nearly immediate, happens after a couple of seconds. And so we decided if that habit has been, quote, broken, will it stay broken? So we did this original experiment, and then Kyle and I, after a few days or more days, would try this again. We never moved the optogenetic fiber, and this is what we saw. And then after 2 to 3 weeks, we did it again, and what happened was that original habit was right back. Now you can imagine we were we were confused. How could it be right back? We've gone on, I don't have time to tell you, and made some very careful measurements of the behavior. And what we think is going on during this time after the habit has been broken, eventually the animals develop a new, they call it replacement habit in the jargon, a replacement habit of going the other way. So pretty soon they're just tooling down, for example, to the right side of the maze. Of course, change the sides. And that second light broke the second habit, but the old one was still there. So now, in our very latest experiment, what we've done is we've said, well, if that's true, it may be that if we perturb every day, not after they've formed a habit, but after this initial training period, when we know that this bracketing pattern has not yet formed, the habit is not yet crystallized, how about every day when they go into the little maze you just saw, every day we do this online inhibition, day after day after day. But they are, we're training them. It's just the light is on. Whoops. And what happens is, I realized that I left out the data for length because I didn't want to go over time. What happens is they never form a habit, and I have the data in an afterwards slide. So this means that we can prevent habits from forming, or if a habit has formed, we can block it, but also if we go at it again, we can get a habit back. And we're extremely interested in the fact that we're doing this by perturbing part of the limbic system. And so we're thinking now in a kind of sketchbook way that partly these loops are definitely, they must be evaluating behaviors, and as I said, selecting them. That maybe this bracketing pattern has something to do with chunking those behaviors or packaging them until you finally have a habit. But what really, truly surprised us was that these behaviors really are quite automatic. You can lay the tracer data of one run out over another, and they're almost perfectly aligned once you really have a habit. But there's some part of the brain, namely at least one part in the medial prefrontal cortex, that's monitoring things. So could it be that when we think we're doing something semi-automatically, there's some guy up here who actually is evaluating our semi-automatic behavior and deciding whether to permit it? So that seems to be the implication of what we are seeing, and it seems to be ongoing moment-by-moment monitoring, which has us very, very fascinated. And that raises a question of, well, is there some storehouse with all sorts of habits in there? We turned off the second habit, we got back the first. I don't know how far back, we don't know. These are all very new experiments. But we do know that we have a lot of scripts or action repertoires that we can haul out. If you tell me, put this down, I can do it, and I probably would do it almost the same way every time, etc. So that brings up the question of what about extreme habits? And I know I don't have time to talk about these, so I thought I'd just tell you in words, briefly, a new result. And that new result is the work that Eric Burger and I did while he was in the lab. He just left a couple of weeks ago, where we went after a model of a compulsive disorder. Whoops, I'm sorry. And what we found is that with optogenetic perturbation in another part of the limbic cortex, we can effectively make compulsive behavior go away. So we're very, very interested to continue this because, as I said at the beginning, it's a thrill to be in brain science now with so much possibility to do many things. And we're trying very hard to keep our eyes on the fact that the basal ganglia are implicated in so many disorders. And any little thing that we can do to help is just what we want to do. So let me thank you and thank people who've been so important. I mentioned the names of several people: Kyle, Tara Barnes, Dan Hugh, Yasuo Kubota, Katie Thorne, Eric, and the wonderful foundations that have helped us. Thank you so much.
So we'll have a break now, and why don't we come back at 11:30? It's 5 minutes after the scheduled return time.